Hi, I'm Heather Morse, Assistant Editor of SJ Magazine. In our December issue, we feature Courtney Kronenthal, Senior Research Scientist at Corielle Institute. We're here at Corielle today to talk a little bit about what happens here in the lab. Courtney, can you tell us a little bit about what you do here?
Sure. So we're standing in Corielle's Genome Center, and here we are trying to understand how we can use DNA information in our healthcare. So this is possible because in 2003 we understood all the chemical letters that make up a human being. Well, a few years later, 2006, 2007, scientists were able to line up the DNA sequences from individuals around the world, and they realized how unique and how similar we actually are. Well, very surprisingly, humans are more than 99% identical in regards to our human genome sequence, in regards to our DNA. So the very less than 1% has somewhat of a big impact. Look how different we look. Imagine people from around the world. We all look very different. We manifest different diseases, right? And we react differently to medications. Well, the less than 1% of our DNA that is unique to us really influences those factors.
Okay, can you tell me a little bit about how you're using that information on how we're different in healthcare?
Right, sure. So that less than 1% is present in the form of SNPs, a very scientific jargon word, single nucleotide polymorphism. What it means is it's a site in our DNA where we differ, a little spelling change in the chemical letters that make up a human. So these are gene chips, and we use these at Corielle in a research study we're doing to try to identify the uses of DNA information in healthcare. So this here is a piece of technology. This is a silicone wafer. It has 6 million DNA sequences synthesized onto its surface. And those sequences represent almost comprehensively the less than 1% of the DNA that make us unique. So we can analyze that less than 1% on this chip and use it in healthcare. So I'll tell you a little bit about how it works. So an individual needs to provide some DNA, and there's many ways you can get DNA, and one is just through saliva. So individuals in our research study would provide some saliva in this test tube. We can isolate or extract the DNA from their saliva sample, and then we inject it into the back of this gene chip. So now the individual's DNA needs to bind to the DNA that's on this little wafer. So if you look over there, there are what are called hybridization ovens. They're basically warm incubators. And for 16 to 18 hours, this chip rotates around there. It allows enough time for the DNA strands to bind. We then take these chips over to what are called fluidic stations here, and they go through washing and staining cycles. Basically, a fluorescent dye is added to the chip before it's scanned with a laser, much like a CD player or a DVD player. And that information then translates to that less than 1% that makes us unique. And we can correlate that information to healthcare decisions.
It's very interesting. To read more about Courtney and the rest of our women in medicine, please check out our December issue.